Thanks a lot. So as I'm the only pediatric oncologist among you, I would like to give you a brief overview of our specialty in the context of P4 medicine and international collaboration, of course. So as you know, pediatric oncology in general is a success story, and that is mainly based on international cooperation already since the 1970s. But still in Europe, more than 3,000 children and adolescents die from cancer each year, and we kind of came to a plateau in the past years and not achieving the same dynamic in the cure rates as we did before. So we have good and bad stories. On the other hand, pediatric patients are treated in Europe-wide clinical trials. Meanwhile, it's not on the national level anymore, but most trials are Europe-wide clinical trials, all of them academic IITs. And the majority of the patients is included, at least for first-line treatment, into these clinical trials, or at least registries, more than 90% of the patients. On the other hand, we observe a reduction in the clinical trial number that is due to the regulatory hurdles and the high costs that have been implemented by the medical regulations since 2004. And meanwhile, we have more registry, not learning about innovation anymore, than clinical trials where we can ask real clinical questions because it's so expensive for the rare numbers of patients that we can include into these trials. It is important to know that cancer entities in children and adults are different. So in adult oncology, as you know, 99% of the cases are carcinomas in one way or the other. These are only making up approximately 1, maybe up to 3% of pediatric cancer cases, and we have different entities. So we need specific research and funding programs because not everything that is developed for adult oncology really fits to pediatric oncology. We face current challenges. Based on the unique molecular features of our cancer entities, as I said, we need specific research programs. What is the largest bottleneck still at the moment is the limited access to drugs, and in particular drug combinations. Pediatric cancer is no market for pharma. We have to do a lot of this development ourselves in academics, and it's hugely difficult to find access to, in particular, drug combinations if we want, based on a biological rationale, combine drug A from pharma company A with drug B from pharma company B. It's almost impossible. So we need more early clinical trials that really have a biological rationale for pediatric cancer. Based on this, we need also academic efforts in drug development. A couple of years from ago, we would have stated pediatric preclinical model systems are needed. This was addressed by a European platform called ITCC-P4. And meanwhile, we have 400 models at hand in Europe that we can use for preclinical testing. And of course, rare tumors always need European and even international cooperation. In Europe, we have the ITCC Pediatric Clinical Trial Platform enabling efficient setup of clinical trials and also serving as a central entry portal for industry in Europe. We have achieved the Accelerate platform. Accelerate is unique because it guarantees involvement of the 4 stakeholder groups, meaning academia, patient advocates, industry and regulators as equal partners in an annual meeting that is focused on a certain drug development topic. And this is certainly a platform that is of benefit, in particular for our rare cancers. We do have European platforms for outcome research, PICORET, or for radiotherapy quality assurance, the Quartet project. So there's a lot of activity ongoing. Our political achievements are made via SIOP Europe, and the current strategic plan for up to the next year contains detailed action plans for 7 objectives, addressing inequalities. Of course, this is also a problem in Europe. Precision medicine and treatment innovation, understanding tumor biology, teenagers and young adults who have their own requests and requirements, quality of survivorship, learning about the causes of cancer, and of course, as everywhere, big data and artificial intelligence. The current status of our pediatric cancer precision medicine programs is quite advanced. So more than 6,000 relapse samples have already been sequenced, currently more than 1,000 new patients are recruited internationally per year. But if you have a look at the map, there are still major gaps in Africa, in South America, in Asia, and we certainly have not achieved global coverage yet. If we look into an example of one tumor, neuroblastoma is my personal interest, we can exactly describe the mutational landscape, in particular of relapsed neuroblastomas, and have insights into the biology of these tumors. But still, we have sequenced more than 250 neuroblastoma patients on our precision medicine platforms. This is the German one, Inform, hosted in Heidelberg. And this gives us 52% targets with high or moderate priority. But nonetheless, in Germany, only 4% of patients are in clinical trials because there's not a fitting clinical trial for every target identified. In France, it's 28% of patients in clinical trials or registries, also including compassionate use registries. And in the Netherlands, it's 24% match targeted treatment if there is a target. But if we look further into the data, cure is achieved only in single patients. And one reason, of course, is that with monotherapy in a targeted fashion, we will not cure the majority of cancers, that's clear. But also, maybe we are not precise enough in our input. So I would like to see it this way. We do molecular characterization, but it's still in the precision medicine programs based on bulk sequencing. Maybe that's too imprecise to identify the drug targets, and maybe that's then also imprecise selection of targeted therapies. So if we have a look into spatial tumor heterogeneity and tumor evolution, these are major problems. We had a study a couple of years ago where we analyzed 5 different samples of the same primary tumor and came up with up to 35% specific mutations in these tumor regions, giving us a problem how to choose the right input for our targeted therapies. So maybe in the future, we should base it on spatial single-cell sequencing to get a real spatial idea of our tumors and have a more precise input of drug targets. And of course, this also needs to be accompanied by drug testing in patient-derived models, like the ITCCP4 European platform, and by therapy monitoring with liquid biopsies, and also here European activities are ongoing. Mona Lisa is a first pragmatic clinical trial for early relapse detection, where we assess the input of liquid biopsies time-wise, and from early data we can already suggest that we will be able to identify a relapse about 3 months earlier than we can do by imaging and other clinical established technologies. So why would you be interested as adult oncologists in pediatric cancers at all? Well, of course, children are the future of our society, so we should be interested in them. But pediatric cancers are also great models for cancer research. Due to their low genetic complexity, it's easier to discriminate genetic drivers from passengers, so we can do great data science in our field. Then we have the assets that more than 95% of patients are in clinical trials, at least for first-line treatment. More than 90% of the biomaterial is collected in national or European biobanks. We have a long tradition of European and international collaboration. And to be honest, rare in your sub-entities is now the new common, so maybe we can learn from each other. Some of the great success stories in cancer came from pediatric oncology, such as CAR T cells. You remember the story with this girl. And I apologize for showing these pictures to non-medical people, but innovation in personalized medicine often starts with individual patients. It's not always about the critical mass. This is a little girl suffering from a very aggressive fungal infection that is normally lethal. We try to control this fungal infection, so the fungus is making holes into the skin and leaving necrosis and looking like a Swiss cheese. So we were successful in controlling the fungus, but this girl only had 2 weeks of acute lymphoblastic leukemia treatment, and giving chemotherapy to this girl would have killed her definitely. So we were in a very delicate situation, what to do, and had to ask the question, can we cure acute lymphoblastic leukemia without chemotherapy? And yes, we can. We gave this girl 5 cycles of blinatumomab, with this a bispecific monoclonal antibody, And meanwhile, this is 3 years ago, her fungus was controlled by 2 years of aggressive antifungal treatment, and she only got ALL maintenance therapy with very low dose methotrexate and purinethol for after many months of immunotherapy. And so this is the first cure of pediatric ALL without chemotherapy, and meanwhile we have 2 more patients to share. So sometimes innovation starts from a single patient and doesn't necessarily need critical mass. Finally, I would like to share Safer Ukraine as an example for global efficiency and for international cooperation. When the Ukraine war started, we got great support from St. Jude Global, and we could set up evacuation logistics within 1 week in 29 countries with 205 participating pediatric cancer centers. So if there is sufficient funding and if there are structures that you can rely on, we can be very fast and very efficient in achieving our goals on the international level. And by this, I thank you for your attention.